Welcome again to Bio-Computation with Max. Last time we talked about biodivergence of COVID, the intensive biocomputation that is being conducted by all of the variants. So now I wanted to discuss some of the statistics. Um, it's not an easy topic, but one that we must think about. So looking at mortality on the global level, you can see on this website that the trend has been up, but we can easily subdivide it into two regions with linearity in each, whether we go with the reported data or the total, which takes into the account the underreporting. So here's the picture. At roughly 7 month time point, there is a break, and the slope turns up, not down. So we're about 14 months or so into the large scale pandemic. So for the first 7 months and the next 7 months, we can look at the slopes and the number of fatalities. And basically, the slope increases by 81.5% for the reported numbers, here from this part colored green to the red dash, or 87.4% from the inferred total, taking into the account all of the underreporting that happens in various countries due to comorbidity or just challenges with compliance or remoteness. In either case, this is quite a significant pickup, unfortunately, and it begs the question of vaccinations. So the vaccinations started roughly in December last year. Israel led the way, and they've vaccinated over 60% of their population very quickly. There are some small countries that are even more vaccinated, but on the global scale, there is no evidence that vaccinations are depressing this rate yet. We hope this will happen, but this evidence is not there. In fact, if we did not know when the vaccinations happened, we would hazard a guess. There are variants that escape the immune system. This is a known scientific fact. So this is perhaps to be expected. Here's a scientific publication from The Lancet in late March. It's talking about the fact that they're able to, a South African variant is able to evade all kinds of vaccines as well as convalescent serum. You saw some of these other designations of that variant. So this is definitely a very real concern. But I highlighted this most critical statement that repeating clinical trials for each variant might take so long that even newer variants could emerge while these clinical trials are underway. This is precisely the challenge of biocomputation that we're facing with respect to COVID. Are we able to modify our response in time? Vaccines may struggle to keep up. This is a fact that we are going to discover in the next 6 to 9 months. Are they able to keep up? How problematic is a reinfection? How severe it is? But this is certainly entering the scientific thinking. In fact, is this a chasing kind of project? Here's another scientific publication and a popular article talking about it by Dr. David Ho, who wrote about these variants, including the South African one, and its ability to evade. And again, I wanted to cite this popular article for this quote, that if the rampant spread of the virus continues and more mutations accumulate, then we may be condemned to chasing after these variants, similar to influenza. That's the seasonality that I mentioned in the previous biocomputation discussion. Time will tell. In terms of ability to predict, well, it's actually quite predictable. So on our website, Fluids for Life, we wrote about this in late March. So because we're all in this learning environment, there is this post I titled Virology 101 talking about antigenic drift of COVID-19. There are other posts that you can see on other websites. For example, our thought leadership website, Bio-Computation. That's the subject of these discussions, as you know. And I wanted to point out the first few of the articles, the need for investment in tools for biocomputation and how pandemic is a driver of that need and should be a catalyst for this investment, and how microphysiological systems could accelerate testing for COVID-19. That's also mentioned in the post here. Specifically, the challenge that we were pointing out is this competition, which is a challenge that it's not clear if we're going to win. The competition is between the ability of the virus to do biocomputation and our ability to chase it with antibodies or vaccines. There are other treatments that may be broader acting, but certainly this issue of antigen drift and virus mutation is a major challenge for both antibody and vaccine, which of course are treatments, which of course are intimately linked. This is an early picture of the supernova of divergence from the early days, showing the split between 20A and 20B. And we see some of the follow-up branches. In terms of computations, there are vast numbers, and you can do the math based on the infected a number of people, as well as the number of particles produced. This simplistic model does not account for long-term persistence in the certain individuals, which is actually critical to evolution of the virus. What would seasonal coronavirus look like if we are unable to keep up? Well, it would be quite more challenging than influenza, but we may be confronted with this reality because the brilliance of the biologist is limited by the tools. So in certain ways, in terms of biocomputation, we have very clunky ways of handling genomics or diversity. So it may be an abacus versus a kind of distributed supercomputer. Let's hope that this is not borne out, but it is a strategic risk that we are all discovering. And the purpose for investment in microphysiological systems is to facilitate this type of biocomputation and utilize human in the dish systems rather than large numbers of folks who are essentially participating in a very large clinical trial. In terms of the viral ability, I mentioned previously the divergence and also this explosion, but there is also convergence. convergence on solutions that are efficacious. So here is a snapshot of a table from Centers of Disease Control website that talks about substitutions of therapeutic concern. And what's notable is that they are prevalent in large fractions of the population and arise in are present in many regions, and in fact arose independently. And some of them actually have analogs. In fact, not only is glutamic acid to lysine, this 2-charge mutation, which vastly affects the binding of the spike to ACE2 receptors, the angiotensin receptors. This double charge mutation has vast effect on binding. But there is another one, which is a single charge mutation from glutamic acid to glutamine. So it's the same shape of the molecule. It's just has a amine instead of an acid group. So this is a convergent solution that has been also found in even more regions, and in fact, in many ways, is more varying. But there are others that are not mentioned here. So this is the kind of search that we could also test, and this is perhaps the gain of function research on some of this mutagenesis that may have been undertaken with or without US funding in Wuhan. But now the virus is searching the space, and it has a lot of foot soldiers. Lastly, I wanted to mention an area of particular concern. It's not just the inefficaciousness of the vaccines, it's the possibility that the breakthrough cases could be severe. The nature of immunopathology of COVID is that the immune system attacks the body, and that is what leads to most severe cases, hospitalizations. Ultimately, the efficiency of the vaccination program will depend on not only the ability of folks to evade reinfection, but the severity of the disease. Fortunately, reinfection or breakthrough cases is exceedingly rare, about 1 in 10,000 for the United States, for example, so far. But there is a very substantial rate of fatality in those cases. So globally, the rate of fatality is about 2.1%. In many countries, like Israel, it had long stabilized at about 0.7%. United States also is at this same global average, but this is a significantly higher fraction. Fortunately, it occurs infrequently, but the question that is facing us is how will the new strains be recognized? If they're not recognized, the immune system will, with specificity, will still become incensed due to vaccination, just as it does with repeated shots of the vaccine. And in fact, that's how it may lead to more severe cases. That has been the prevalent discovery when vaccines were being developed for SARS-CoV-1, the first variant that emerged in 2002-2003. So let us hope that this is not the case. Thank you.